Molecules can be described as inorganic and organic molecules. Most of what we have inside of cells and living organisms is going to fall under the organic category. Organic molecules, by definition, are molecules that contain carbon and hydrogen. So things that would not be considered organic molecules would be something like water, because water is H2O. So notice there's not carbon and hydrogen in that molecule. So that's on the organic side. Another one would be something like carbon dioxide, which is CO2. So again, there's not hydrogen in that particular one. But we will have lots of organic molecules. One of the reasons for this is because carbon had that ability to make 4 different bonds surrounding it. So that could be 4 bonds to 4 different things, which gives us a lot of variety. The type of molecules or just examples of molecules that we would have on the organic side would be proteins. We have carbohydrates. A lot of fats would fall under that category as well. So we are going to talk about some of those, just the general structure of them and the general properties of them. But as we start to talk about these, they are not just going to contain carbon and hydrogen. They will have some special bits and pieces to them that help to give them their unique functions and ultimately the names that we do refer to them as. Functional groups will provide these unique characteristics to the organic molecules, and again, they help to define these groups for us. Here is a table that goes through and lists a number of functional groups that we do find commonly in organic molecules. You've got the structure of them shown in red, so you can see that they do vary in what they actually contain, the name of them, and then in the last column of this table, we can see what they actually are found in, so what type of molecules. So just to point one or a couple of them out, this one right here, it's called a carboxyl group. This carboxyl group is going to be found in amino acids, it's found in proteins, it's found in fatty acids as well. So it's a pretty common functional group or just chemical arrangement that we do find in organic molecules. And then one on the bottom here, the phosphate group, this one has got a phosphorus in the middle and then some oxygen surrounding it. That one is found in phospholipids, which are very important for cell membranes. It's also found in ATP, which is really important when we do talk about energy involvement in chemical reactions. Common organic molecules, as we mentioned, are going to be lipids, carbohydrates, proteins, and then also nucleic acids. So let's talk about some of those different molecule groups and what their properties happen to be. Lipids, as a group, are extremely diverse, and what's going to kind of tie them all together is that they're not going to dissolve in water. So you're probably familiar with what you're seeing here in the beaker. So we can put oil into water, and that oil is not going to dissolve. It stays in its own little droplets or in a layer on the top. That's an example of a lipid, and that's specifically an example of a fatty acid. Fatty acids, they do have a special functional group on one end, and that's the carboxyl group that we were just looking at. And then they're going to have this long chain that's just hydrogens and carbons. And so those are fatty acids, a couple different types that we do have inside cells. We have what we call unsaturated fatty acids and also saturated fatty acids. And these fatty acids, they can be put together to create some larger molecules that are also called lipids that we're going to find inside cells. If we look at some of those other lipid molecules, here would be the triglycerides. Triglycerides are also commonly called fats, and these take essentially 3 of the fatty acids, so 3 of the structures that we just saw, and they're going to combine them together. Notice that when we combine them together, it's a dehydration synthesis reaction because it is a synthesis or a making reaction. If we were going to make this, we would have to actually use some energy to make that happen. So a fat or a triglyceride is just a really large lipid. Another lipid group is the phospholipids. Phospholipids actually take 2 fatty acids, and now they're going to take a phosphate group and again combine all that together so that we get something a little larger. Again, since we're combining things and we're making something more complex, this is going to be something that requires energy. Now, when we look at the phospholipid, we end up with this molecule, and you can see it described here, kind of summarized right here, where you have a portion of it that's very hydrophilic, or in other words, it likes water. So water will want to surround that particular area of the molecule. And then you have the bottom portion, shown here in yellow or tan, that does not like water. And so water won't have anything to do with that particular region of the molecule. And we're going to find that phospholipids are really important for cell membranes, and they will associate together to form a nice barrier around the outside of the cell. We'll also see them surrounding some of the inside compartments of the cell as well. So if we look at how they're going to assemble together to actually create membranes inside the cell, this is what you have, where you have basically a whole row of them here, where all of the ones that don't like water are stuck next to each other, and the parts that do like water are stuck next to each other. And we end up with what we call a bilayer, and that bilayer, bi means two, so it's basically two layers of these phospholipids that are associated together to make this nice barrier around different cellular components. So if we leave the lipids, many of the other molecules that we find inside the cell, they're going to be really large, and they're going to be what we call polymers. Polymers, poly means many. These are large molecules that are formed by linking many smaller pieces together, and those smaller pieces that we're going to connect together, they are called monomers. Now, depending on what the structure of the monomer is, we end up getting a different polymer at the end. Common polymers that we do have are carbohydrates, proteins, and then also nucleic acids. If we talk a little bit about the carbohydrates, if you just break down the word, it's carbon and hydrate, which is water. They are going to contain carbon, hydrogen, and oxygen in the ratio of CH2O. So basically water for every carbon atom that you happen to have. Now, carbohydrates are important to us, first off, because they are a major part of our diet. If you were to look at the food pyramid, pretty much these two bottom tiers right here are all carbohydrates. So this would be divided into what we like to call complex carbohydrates, which are a lot of things that are on the bottom. So breads and pastas and potatoes, things like that, grains. And then we also have the second layer, which is going to be more what we refer to as simple sugars. That would be a lot of your fruits and vegetables. So carbohydrates, they are a major food source for us. They're a major food source for us because our bodies, our cells, are going to break those down and get energy out of them. We would get energy out because breaking is going to be one of those decomposition reactions. The carbohydrates come in three sizes. We have monosaccharides, disaccharides, and polysaccharides. And the big difference between them is the prefixes. Okay, so mono means one, di means two, poly means many, and this is just describing how many pieces have been linked together. The monosaccharides are obviously the smallest. We have some examples of them right here. Sometimes they have a linear structure. Sometimes they have more of a ring-like structure. So we can see both of those configurations. When we start to actually take them and join them together to make disaccharides or ultimately polysaccharides, it's going to be the ones that are in the ring form that get joined together. So here we have a dehydration reaction, so a synthesis reaction that's putting together two monosaccharides to get a disaccharide. And so the disaccharide, if you look at it down here, it has two pieces hooked together. This particular one, sucrose, is one that's very common. This is essentially what you buy when you buy a bag of sugar at the grocery store. So the big 5-pound bag of sugar, that's sucrose. It's a disaccharide. So disaccharide, two individual monomers, they've been linked together using this reaction here. And because we are building, notice that there is going to be energy going into this reaction or being required for that particular reaction. So both of these are what we like to call simple sugars. Simple sugars are very easy for our bodies, our cells, to actually break down and get the energy out of them. The polysaccharides are going to be much, much bigger because these are going to contain thousands of individual monosaccharides that have been joined together using those dehydration reactions. So they get huge. Here is just showing you from a distance, kind of really zoomed out, some different polysaccharides. So we're just seeing little rings that have been hooked together. Examples, common examples, starch. Okay, that is what's in a lot of our food. So that's an example of a polysaccharide. Glycogen is a polysaccharide that's produced inside a lot of bacteria and then animals. And then cellulose is one that we do find in some cell wall structures of plant cells. It is a very fibrous structure, and that is what is fiber when we talk about us eating fiber. Another important polysaccharide is going to be called peptidoglycan, and that's one that we do find in the cell walls of bacterial cells. So basically we have some polysaccharides that are for structural purposes, so for building rigid structures like cell walls, and then we have others that are used more as a storage form for energy, and that would be your starch and your glycogen. So the ones that you see here, we do call them complex sugars because they are so large, and because they're so large, it is going to take more time for sure to break them down and get the energy out of them. Proteins are really important to us as a food source as well, and they are going to contain carbon, hydrogen, oxygen, and nitrogen, and the proteins make up this layer right here, so nuts and seeds. They're also found in this layer of the food pyramid if we were to look at it. So they're definitely things that we do eat a lot of. They make up a major source of the human diet. And they are going to perform a lot of different cellular functions when we just talk about the ones that are found inside of our cells. Basically, if there is a function that you can think of that needed to occur inside of a cell, then there's probably going to be a protein that's going to perform that function. They help to maintain cell structure. So what we mean by that is that they help to maintain the shape of the cell. We will have proteins associated with the outer coverings of cells a lot of times. They are going to catalyze reactions, and that would be when they are functioning as enzymes. They help to regulate the cell's activities and regulate other molecules inside the cell. So to control different parts of the cell is another way to say that. Another thing that they do is they help to move things around the cell, and they also help to move things into the cell and then also out of the cell. They're also, lastly, important for defense and offense. So antibodies would be a good example of a type of protein that's involved in this defense type of activity. If we were to look at the structure of the proteins, these are also really large polymers. They are polymers made of amino acids. So the amino acids are the monomers, and the amino acids is what we're seeing right here. So this is the amino acid. It itself is a relatively small molecule. It has two different functional groups on it. We've got the amino side, which is the nitrogen side, and then we've got the carboxyl side, which is the side that's got the carbon with the double bond to the oxygen and the OH. Our proteins are made of 20 different amino acids, and those 20 different amino acids will have something different in this particular location. On the bottom of the slide, we've got 6 different examples of amino acids listed. So those 6 are just 6 of the ultimate 20 that we have possible. But notice that the R component, which is the component that's shown in the blue, so the blue shading right here or right here, for example, that part is different among them. The other portion is going to be exactly the same the whole way across. So these amino acids, they're going to be ultimately connected together with some synthesis reactions. And when they are connected together, a peptide bond is the link that we're going to find between them. So here we see 2 of them being joined together to make one that has 2 components, and we call that a dipeptide. And eventually, we're going to hook a whole bunch of them together, probably somewhere along the lines of a couple hundred to a couple thousand. This is what we call the primary structure of the protein. The primary structure is the starting point, but one thing that's unique about proteins is that we don't just have to hook everything together. That long molecule has to be very carefully folded up to make this very stable, perfectly shaped 3-dimensional structure. So that means that our proteins require special folding. And that special folding is absolutely required for their ultimate functioning inside of our bodies, inside of our cells. So after we get everything linked together, our cells take those proteins and they put them through a secondary level of folding. We call that secondary structure. With the secondary structure, we end up getting what are called alpha helices, where they actually get spiraled around. Some portions of it are going to get folded into a rigid structure that kind of goes up and down, and we call those pleated sheets. Once we get through that level of folding, then the next level is the tertiary, or the third level. And that takes all of that and starts to fold it up even more so that we have a nice three-dimensional structure. And then sometimes that one has to associate with another one, and that's what we call the quaternary structure. So all of this is to mean that it takes a lot of time and really carefulness to get from what we started with just amino acids to ultimately the protein that's going to function for us. There are a lot of different shapes to proteins, which is why we have a lot of different functions for our proteins. Any changes in conditions inside the cell can possibly cause unraveling of this nice, perfect structure. So changes in the environment, if they do unravel the protein, that is going to cause the protein to be non-functional, which obviously is not a good thing. That unfolding or unraveling is what we call denaturation. So extreme temperatures might cause that. Changes in pH might cause that. We don't want our proteins to actually unravel and unfold because, again, it destroys the function of those proteins.